So we're going to be shifting gears here. We're shifting gears into our discussion of genetics. And so in living environment, the genetics that's a part of the curriculum includes some review from what you learned in 7th grade. 7th grade, you talked about a lot of this stuff. Some of it will be reviewed. You talked about genes and Punnett squares and pedigrees. So we're going to sort of review that. We will then take things a little bit further this year than you learned in 7th grade. So when we talk about genetics, genetics is the study of how traits are passed from one generation to another. It's the study of what we call heredity. You've heard the term hereditary. So if a condition or a disease is hereditary, it means it's something that can be inherited. It can be passed down from parents to their offspring. And it does that through the transmission of DNA. Our DNA is where our genes are located. Okay, we inherit our DNA from our parents. It's found on the chromosomes that were fertilized to produce us. So, in terms of history of genetics, there's some famous scientists that studied genetics and added to our knowledge. One of the most famous is Gregor Mendel. And you learned about Mendel, I think, in 7th grade. Gregor Mendel was a monk, a religious person from Austria. He lived in the middle of the 1800s. So he was a monk and lived in a monastery and did his religious duties, but he's also sort of he was a hobbyist, or he was interested in science, especially in growing plants and studying them. So he is sometimes thought of as the father of genetics, because what he did is he bred pea plants together. So like other plants that produce flowers, pea plants reproduce sexually. Okay. And so you can take pollen grains from one pea plant, use them to pollinate another pea plant, and so basically you can force them to breed together in whatever way you want. And what Mendel did is he did this thousands of times, taking different types of plants with different characteristics, pollinating them, and then taking very careful notes, careful records of what happened when he took this pea plant and crossed it with this pea plant. What were the offspring like? What traits did they have? And so this is Mendel in the lower left-hand corner. This is his notebook. And, what really Mendel does is just kept very careful track of all of these different plants that he was experimenting with. And what he noticed is that he started to see some patterns to the inheritance of certain traits. Mendel was looking at things like the color of the flower, the color of the pea, whether the pod was smooth or wrinkled, whether the plant was tall or short. So all these different factors he was looking at. And he saw that there were certain patterns to be found. And he established the 3 main laws of genetics that we're going to talk about. So, interestingly, though, his work was kind of forgotten about for a while. So he did publish his work, but it wasn't really that widely read. And it kind of fell out of the record. But then as scientists started studying inheritance more in the 20th century, some scientists sort of rediscovered Mendel's work and put that together with the work they were doing to better understand genes and genetics. Thomas Morgan is a famous geneticist as well. He came up with the term gene. He did his experiments on fruit flies. So kind of like Mendel, these first experiments were done with relatively simple organisms, the pea plants. Fruit flies are relatively simple in that they have some traits that are easy to observe, like red eyes versus black eyes, like the wing shape. And fruit flies reproduce really quickly. And so you can study them. They're small. You can raise them in a lab. So they're an organism that can be experimented on and studied pretty easily. And what Morgan did is he sort of saw that when he was breeding these fruit flies together, they were following the same patterns of inheritance that Mendel saw in his pea plants. So this allowed scientists to understand that, well, Mendel's laws aren't just about pea plants, they apply to animals as well. And that was a big advance. And there's lots of other scientists that learned about genetics. When we start thinking about DNA, probably the most famous scientists are Watson and Crick and Franklin. So James Watson and Francis Crick, they were geneticists that were studying sort of what the DNA molecule was like. And at the time, scientists weren't sure about the structure of a DNA molecule. How were things connected? How did it encode information about an organism? Watson and Crick were working on that problem. An interesting historical note is that Rosalind Franklin was a woman working also on the question of DNA. She was using X-ray crystallography to sort of study the nature of what the DNA molecule might look like. This is what an X-ray crystallography looked like. And she was experimenting with DNA, and Watson and Crick actually saw her photographs that she took without her knowing. And after seeing the X-ray micrographs that she was producing, that's what sort of clicked with Watson and Crick and allowed them to come up with the correct model of the DNA. But they didn't actually do this work. Rosalind Franklin did. She didn't get the same credit that Watson and Crick did. They won a Nobel Prize. She did not win one for her work on DNA. So there was certainly some controversy, and especially at that time, there were not many women working in science in universities, and so it's an interesting story about how her work was kind of co-opted by these two male scientists that they eventually became famous for. So Watson and Crick discovered the structure of DNA. This is their model that we're going to be learning, or we actually learned about it back in biochemistry. We'll be reviewing it again today. So let's come back here and think about Mendel's work. So Mendel was working with these pea plants. They're easy to grow. They have some easily observable characteristic. Some of the things he was looking at was the shape of the seeds. They could be wrinkly or round. The color, green or yellow. With the flowers, you could look at whether they were purple or white, where they were located on the plant. Was the stem of the plant long or was it short? Were the pea pods green or yellow or smooth or wrinkled? So he's looking at all these various characteristics. Okay, and he came up with several laws about how these traits that he was looking at are actually passed down from one generation to another. So one of the first laws is Mendel's law of dominance. And this is the question I asked you about today in our daily question. What does it mean when we say one trait is dominant and one is recessive? So first off, there's a term we use called allele. An allele is just a version of a trait. So for example, the trait of the shape of the seeds. There's two alleles pea plants might have. They might be round or wrinkled alleles. For the seed color trait, there's two alleles, green or yellow. So allele just sort of means a version, a possible version of this trait. And some alleles are considered dominant, and others might be considered recessive. So when we say that an allele is dominant, it means that it's going to show through if it's present. So if an allele is dominant, it means it's going to sort of overpower a recessive allele. And the dominant allele is the one that's going to be displayed by the organism. When we are using Punnett squares and writing symbols of different alleles, what we often do is use an uppercase letter to symbolize the dominant allele. And we use a lowercase letter to represent the recessive allele. And so when Mendel took pure tall pea plants and pure short pea plants, and he cross-pollinated them, and he grew the seeds that resulted, what he found is that all of the offspring were actually tall. Even though one parent was short, every single offspring was tall. And so what do you think that led him to consider about the tall allele versus the short allele? If tall always showed through in the next generation. What does that mean about the tall allele?
That was the dominant trait?
Yeah, that was the version that was dominant. And the short allele was recessive. So if we look at that, using our symbols, we'll use a T to talk about tall or short. So one of the parents in that pea plant had two big T alleles. We say that was its genotype, was big T, big T. The other parent that was short, it had a little t genotype. So this is a tall plant. Its phenotype is sort of what its appearance is. It was tall. That's its phenotype. This one was short. That's its phenotype. So we use these models called Punnett squares, these little boxes, that help us predict if two parents produced offspring, what would they be like? So, to do this Punnett square, we simply combine the letters to fill in each box. So this top left box, we get a big T from the top, we get a little t from the side. Same thing over here, big T from the top, little t from the side. Big T, little t, big T, little t again. So this Punnett square shows us what Mendel was seeing in his experiment. When he took this pure tall plant, crossed it with this pure short plant, every single offspring was tall. And like Francesca said, that's because the tall allele is dominant over the recessive allele. And each of these boxes represents 25% of the offspring. Every single one of them ended up with a big T in it. Because it's dominant, that's what shows through. Okay. We call these, when we have two of the same allele, that's called homozygous. The prefix homo means same. These offspring are heterozygous. Hetero means different, in that they each have two different alleles. Then what Mendel did is he took two of these offspring and he crossed them. So he took a big T, little t offspring and he bred it with another big T, little t offspring. So let's fill in this Punnett square. So again, this is a heterozygous parent and another heterozygous parent. All right, you guys tell me what these would be. Reagan, what's going to go in this top left box of this Punnett square?
Capital T.
Yep, from the top.
And, yeah, two capital T's, right?
Right. A capital T from the top, a capital T from the side. Cameron, what's going to go in this top right box? Yep. Capital T from the left, a lowercase t from the top. We always usually write the big T first. It doesn't really matter. How about this lower left-hand box? William, what are we going to write in this lower left-hand box?
A capital and lowercase t.
Yep, capital T from the top, lowercase t from the side. And Francis, how about in this last box, lower right? What's going to go here?
Capital and lowercase t.
What's it get from the top, Francis?
Two little t's.
Yep, it gets a lowercase t from the top, a lowercase t from the side. Okay, so what is this telling us? This is telling us that if these two heterozygous parents reproduce 25% of the time, their offspring is going to have big T, big T. 25% of the time, they're going to be little t, little t. And then 50% of the time, they're going to be big T, little t. Each box represents 25%. And Mendel saw this. When he did this experiment and grew the plants, he saw about 75% of them were tall and about 25% were short. So 25% short, this would be tall, tall, tall. And so he saw this after he did it with flower color. And he did it with whether the peas were wrinkled or not wrinkled. And he kept finding these same ratios. This ratio of when you cross two heterozygous parents, you always end up with a 75%, 25% offspring. So why? Why has this happened? So Mendel saw that, or came up with this law called segregation and recombination. So we learned about meiosis recently. And we know that in meiosis, the gametes that are formed, each gamete gets one of the homologous chromosomes. So the alleles that are there, that a parent has, get separated during meiosis. Each gamete is only carrying one allele for every trait. So they're being segregated, they're being separated. And then during fertilization, they recombine. And so an allele for each trait from the mother and an allele for each trait from the father recombine to form the two alleles in the offspring. Okay? And that's what's being shown here. In meiosis, these cells that have two homologous chromosomes together, they get separated. Okay, let's say, this is a gene for brown eyes, and this is a gene for black hair, and this is a gene for blue eyes and red hair. During meiosis, depending on how the chromosomes line up, they get separated from each other. So every single gamete that's produced has a different combination of traits. And so which of those gametes ends up in the offspring, that's going to determine the traits of the offspring. Mendel also saw that the allele for pea color was separate from the allele for whether the pea's wrinkled or not. That the traits he was studying, they were all separated and they were inherited independently. And that's because the traits he was studying were all on different chromosomes. Okay? Now, sometimes if the traits happen to be on the same chromosome and are close together, then they can be inherited together. They are linked. So one idea that Mendel came up with was, if you have a flower that's purple, let's say. Purple is the recessive allele for flower color in peas. How do you know if this purple flower has the genotype? I'm sorry, purple is the dominant color. How do we know if this purple flower is big P, little p for its genotype, or big P, little p? Because both of those combinations are going to give you a purple flower. So how can you tell which it is? The way you do that is using a test cross. In a test cross, what you do is you take this dominant individual and you cross it with a homozygous recessive individual. A white flower would be in this case. So if you breed this purple flower with a white flower, then you look at the offspring. Because if this purple flower is big P, it's homozygous, are you ever going to get white flowers as a result? No. All of the offspring are going to get at least one dominant allele from this plant, and all of the offspring are going to be purple. However, if you do that same cross, you cross this purple flower with a white flower, and you see that some of the offspring are white and some are purple, then that can tell you this purple flower must have been heterozygous, because that's the only possibility that ends up with some white flowers. So this is what a test cross is. Any questions about our notes so far? Okay, I want to show you this video to sort of bring all the things we talked about together. You can see some animations and so forth that's going to help you to understand this.